I'm the bat bodyguard, and I'm here to protect bats. Up here, you can see a bat. Up on the left, he's got some white fungus all over him. I have some proteins in the center, kind of hard to see, like gold background. And over on the right, microscopic view of some fungus. Kind of lays out what my whole project is biochemistry. Specifically, I'm working with a disease called white nose syndrome. It's found in hibernating bats that are cave dwelling, and it only occurs in the winter. What happens is this fungus goes on sleeping bats, covers them in white fungus, especially their noses, and it wakes them up, and it wastes all their energy in the winter when they should be sleeping. Imagine if you were sleeping and you got woken up every single night, wasting all your energy for 3 months, you would die. It actually kills 90 to 100% of infected bats. So it's a very serious problem. Serious enough, it started in New York about 2006, and it's been coming west ever since. It's actually now in Hays County in central Texas over the last year. San Antonio has the nation's largest bat colony, millions of bats. And this winter, if this is not stopped, millions of bats are projected to die from this white nose syndrome. This is our villain, Pseudogymnococcus destructans. I'll refer to it from PD from now on. Some key facts about it: it loves cold temperatures, about 15 degrees Celsius. It affects hibernating bats and only bats, so don't worry about humans. We're not going to die from this. And it also needs copper for survival, just like every other organism, we use copper. But I mention this because a lot of the rationale behind my project is focusing on copper trafficking proteins. Copper is really important. It can do things such as be present in superoxide dismutase. Many enzymes in our body need them, including like eukaryotes like yeast. There's a diagram here, and it kind of shows how copper is brought into the cell. Many of you have taken functional biology, and you're aware of transporter proteins that bring copper in the cell. Well, I'm not focusing on that. I'm focusing on proteins that bring copper to the transporter protein. So it's kind of like a big linkage. These BLPs, we call them BIM-like proteins because they're kind of like BIM proteins that are in another fungus. There's a lot of investigation there. So these are uncharacterized proteins that we call BLPs. They grab onto copper and bring it into the cell. And our goal is, if we can somehow stop these special proteins that are only in this eukaryote, maybe we can stop the growth of PD just a little bit. Even 10% would be enough to let the bat survive. So just a minimal growth, we can stop that. And this goes on to my project. What am I actually doing? Well, first, I'm taking genes from PD and making recombinant DNA vectors. So I'm making my own new plasmids. Then I'm going to put these plasmids in a bunch of model organisms that I'll show you later. I'm going to purify some proteins. After I verify it, I can actually express them. Do a lot of SDS page gels. Affinity chromatography. I'm using stuff like his tags, strep tags. Biochemists are aware of these, what they do. Very high affinity. Once I can actually have the proteins, I can move on to studying their biochemistry. What do they do? How do they react to copper? How do they react to chelators, stuff that grabs copper and keeps it from ever coming into the cell? All the meanwhile, I'm going to try to isolate RNA from the actual pathogen itself, PD. If you isolate RNA, you can do transcriptomics, and you can see which genes are being upregulated or downregulated. So you can see all my proteins of interest actually being transcribed. Here's some of my model organisms. There's 3 of them. I didn't always have 3. E. coli hasn't worked. Hasn't proved too valuable in actually expressing the protein. But I'm still trying. I got the special shuffle T7 cells. They help chaperones to help fold the protein. And I have 2 yeast, P. pastoris and S. cerevisiae. These are yeast like the pathogen, and so they're really good at actually making the protein I want in its natural environment. You'd be surprised how a yeast protein doesn't survive well in a bacteria. So These are some genes I made from plasmids. I just put a bunch of stuff together, they ligated. Some of my results, the past month I've been doing a lot of cloning. Basically, I take constructs, I ligate them together, and I verify if I actually have them. So all my E. coli ones I actually verified, with my gel on the bottom left. My P. pastoris I also verified. Some of my first gels I ever did, so I had to really overexpose them to see the correct bands. It's just part of the learning process. And I'm still working on some of the cloning for Escherichia, trying troubleshooting the cloning in our lab, a couple of bad reagents. But moving on to expression, on the bottom left here, my E. coli, the red box shows a band, and that band is for my soluble fraction. So there's some soluble protein. When you're working with proteins, they gotta be soluble, otherwise you can't use them in solution. But it quickly goes insoluble, so it's kind of like limited stability. I probably can't study it. That's why I'm using yeast. And the red box on the P. pastoris gel, that shows I'm actually expressing some proteins. They're all glycosylated, so I had to cut them with some enzymes. And that's where I'm at now. So for my future directions, I'm hoping to optimize my actual protein expression. I'm changing how long I express them, the temperature, the media, what kind of inducers I'm using. All these different troubleshooting. It's pretty fun. I'm also hoping to actually do some in vivo experiments with the pathogen itself. Long term, all that biochemistry I talked about, it goes back to white nose syndrome. So white nose syndrome, it's a bad syndrome. And there's no treatments at all. So there's no cures, no treatments, zero. No one's doing anything to do it. So if I can learn more about how this copper proteins are bringing in copper, maybe we can stop it and inhibit growth just a little bit. So that's the goal. Hopefully we can provide some treatment and show how to limit pathogen growth. That's all I have. Here's like the lab picture of my few lab mates. Back during COVID, we're always social distancing. Any questions about my project or the pathogen or bats or anything at all? Yeah, goes away.
I got confused whether you were studying the proteins in the bat or in the pathogen. And how does the protein, what is the cause of the fungus, why is it growing, what nutrients is it taking from the bat, and and why the copper?
Yeah, a lot of fantastic questions. So here's the bat itself. I'm not studying the bat. There's no bats in my lab. I think it would be pretty unethical to use the pathogen or the bat. I don't want to hurt any bats. I don't want them to die. So I'm taking proteins from the pathogen and putting them in new organisms I can study. So I'm not studying the bat. Often the pathogen will go into the bat and just suck up all its energy and kind of slowly eat it. The pathogen is pseudo-germicomposite destructions. It kind of needs what you and I need. It needs water. It needs some carbon source, some food. It needs copper, just like we need copper. But it grabs copper in a very special way, different than us. So we can target how it grabs copper differently than how the bat is grabbing copper. So we won't kill the bat when we spray it with the drug. So it's very important to have differences in how to treat them. Does that answer your question a little bit about what I'm studying?
Yeah, you were studying the fungus.
Yeah, that guy right there. The genes in that.
Yeah, how it works.
Yeah, that's all you.
Does the type of bat matter? Like, does it impact a certain species of bat more than others?
Yeah, absolutely. So this fungus is really lazy. It doesn't have any legs. It kind of just sits there. And it only grows in the cold. So therefore, it infects bats that hibernate, and it infects bats that also are cave-dwelling. So some bats that are in trees or in other locations are unaffected. But if they're in caves and it's the winter and they're hibernating, that's a perfect storm. So it's several species, like tricolored, small bats. Yeah. Kaylee?
Oh my god, did I forget it? I think I forgot it. Oh, I remember. You were saying that you were that be like mice or like something else that's more comparable to like what a bat would be? Or-
Actually, we're using these organisms.
Oh, okay.
So we're using P. pastoris and S. cerevisiae.
Got it, okay.
S. cerevisiae is actually baker's yeast, what's used to make bread. Very well studied, and therefore we can put the pathogen genes in that organism,
Got it.
and it acts very similarly to our pathogen. And you can study the biochemistry. You know what's going on with like the Golgi apparatus, how are proteins being glycosylated, how are they picking up copper. So I won't be actually touching the bat. I'm looking at the pathogen, because the bats are going to survive no matter what, but I want to stop the pathogen from infecting bats. Yes.
So wait,
And
go ahead.
that's all your question.
So I'm trying to think of a way to say this in a way that doesn't insult the bats, but they're not the most popular animal right now. Like I'm sure that save the bats is harder than save the sea turtles. So how do you sell bat conservation? Do you take like a pollination route or?
Yeah, I think that's a question. I wanted to break that up too. I almost forgot to.
Well, like would you bring this like agricultural groups, do you think?
Yeah, there's actually a white nose syndrome group
Yeah.
with like hundreds of team members. So I'd likely bring it to them.
Okay.
More likely a scientific journal, honestly, and they could use that information.
Yeah.
There's been some papers investigating how pathogen responds to certain metal chelators, but no specific experiments. So I'm the first to actually take a metal chelator or take copper to the pathogen and see what happens. No one's ever done that before. I'm not a biologist, so I'm not outgoing to conservation groups, but it's important for grant funding to talk to those groups and say, hey, here's the biochemistry, here's a drug, I can give it to you, and you can implement it and make new guidelines on how to use it. Here's how the copper works and the enzymes and the ligands. Yeah.
Cool.
I was wondering how, after let's say you develop a drug, how would you administer the drug to thousands of bats?
Oh yeah, that's not too bad actually. I wouldn't even develop the drug. It's just a simple chelator. Some of them are called, let's see if I, yeah. So down here, BPS and BCS. These are just molecules that love to grab metal ions and hold them from going into the cell. I call them chelators. All you gotta do is like put that in a bottle, and you can spray it at the right concentration, like a spray bottle. Like, you just spray all the bat colonies, just enough to where it doesn't hurt the bats, but it inhibits the growth slightly of the pathogen. 10-20%, the bats will survive the infection. So yeah, you can just spray them. This is one possible treatment. We're looking into a lot of other chemicals. They're like dandruff, the shampoo chemical that stops dandruff, like it's like zinc, pyridazole. There are chemicals like that that are already made. So we're not developing drugs. We're kind of just investigating what can we use on this pathogen.
Yeah, thank you.
Yeah. Any last questions for the bat bodyguard?
Where'd you get?
And one last
Sorry.
question for Daniel.
Daniel?
Have there been any cases of like other cave-dwelling hibernating, like for example, like a bear getting hit by it?
No other species but bats. It has to do with bats have some slimy stuff all over their wings actually, and it sticks really well to the fungus. And plus it has to be like cold and like wet. So no other animals. It's just been bats so far in caves. We thought it came from Europe originally on like cave exploring equipment, and it came to America. It's actually an invasive species. It may go down to South America eventually. Now it's in Texas. So it's really a big problem in the last decade that no one really knows about, except you all know now. So.
Okay.
Last full comment. Yeah, what do you have?
Where did you get the magic card? Is that what that is?
Oh yeah, so I have this up here.
I love that.
Yeah, so I made this little, it's like a trading card that has the pathogen on it. Yeah.
Did you make that or did you?
Oh, I made it. Yeah.
You made that?
Yeah. He's a bad guy. Yeah. And you can't grow it like that. That's like a lie. That is so hard to do. This thing is very hard to work with. That's why I have other organisms. But I'll stop at that. Hope you guys like the presentation. And that's all for now, yeah.
Great job, Chris. Thank you.